Hi, everyone. My name is Jessica Giordano, and I'm a genetic counselor in the Department of OBGYN at Columbia. I want to thank the organizers for the opportunity for myself and Dr. Ron Wapner and Dr. Peter Robinson to share our views on a vision of genomics across the perinatal continuum. Today, we're going to go through three different objectives. First one is going to focus on describing how sequencing in the prenatal period helps to better inform the postnatal management. We're also going to identify necessary data collection in perinatal genomics, which will allow for a more robust, efficient, and accurate diagnosis and overall improve clinical care across the perinatal continuum. And then lastly, we'll touch on opportunities for prenatal genomics and the newborn screening stakeholders to collaborate further. So first, I want to focus on perinatal sequencing and the impact on management. It has now been pretty well described over the last number of years that rapid sequencing in the NICU can very much affect and direct medical management. And in this paper from 2017, where they sequenced 278 critically ill newborns in the first 100 days of life, they made a diagnosis in 36.7%, and they actually found that 38.2% had an atypical or unrecognized presentation of genetic disease. That represented some kind of phenotypic expansion or some heterogeneity. They also noted that diagnosis affected the medical management in 52% of cases, and that included a redirection of care, a reduction of the diagnostic odyssey, potentially an initiation of a new subspecialist care or additional diagnostic studies, changes in medication or diet, or the start of major procedures that were necessary. And from my vantage point as somebody that routinely analyzes prenatal genomes in the setting of congenital anomalies, I very much noticed that there's a significant overlap between the prenatal diagnoses that we see on exome and genome sequencing and those that are actually made on baby sequenced in the NICU in the setting of congenital anomalies. But I would like to argue, and I think the data is really beginning to show, that prenatal sequencing informs pregnancy and delivery management, as well as the immediate management right after delivery for the fetus and the mom. And so a review by Kingsmore noted that the rapid, or quote unquote rapid, neonatal sequencing that's occurring across the country has a median turnaround time that ranges from 9 to 23 days. And I would argue that that may not actually be fast enough for some of our families facing diagnosis of congenital anomalies. And I would actually say that the most rapid NICU sequence is really the one that's obtained prenatally. And we're starting to see this literature really come to light, and we're currently working on a large multi-center study to try to further identify the exact benefit of prenatal sequencing versus postnatal evaluation. But in this very recent review by our colleagues at Cincinnati Children's, just published 2 months ago, there was a retrospective review of 20 cases and that had congenital anomalies in prenatal exome sequencing, and they noted that there was a clinical management change in 45% of the patients. And you can see those diagnoses in the chart, and these changes included changes in family planning, neonatal medical management, as well as diagnosing additional family members. I would like to also point out that there are benefits of prenatal sequencing when there are negative results as well, and specifically that the diagnostic yield does not directly equal the clinical utility. So there's 2 studies from the Netherlands that reported that clinical management was directly impacted by prenatal exome sequencing in 70% of their patients who had sequencing. And those changes included using the results for pre and postnatal medical management. Of their 43 cases that had negative prenatal exome, about half of the patients had clinical management changes primarily related to supporting their decision to continue their pregnancy or pursue intervention for the fetal anomalies. And just to further that point, I want to use the example of a fetus that has an apparently isolated hypoplastic left heart syndrome. And the parents are considering during their pregnancy whether they want to pursue palliative care or postnatal surgical repair. And if their exome result is positive, parents may be concerned about quality of life and opt for palliative care. And if it's negative, they may feel more optimistic about the outcome and opt for postnatal surgical repair and possibly transfer their care to a hospital system that has pediatric cardiac surgery. So there's a wide range of benefits of doing sequencing prenatally from what we've seen in the last number of years. We're seeing improvements in management, changes in delivery site, selection in the type of delivery, potentially in utero management and treatment of a fetus, and that is slowly starting to expand, as well as reducing the diagnostic odyssey, making plans for potentially palliative care, and we allow families to maximize their reproductive options. And so our goals in prenatal diagnosis, I think, really align with the goals of those that are working in the newborn screening space as well. We're all really working to improve the health and management of newborns and their families. And early prenatal diagnosis may result in prenatal intervention that can improve those perinatal outcomes and the immediate care of the newborn. It can allow for delivery management to optimize newborn and infant outcomes, as well as more education of parents regarding the special needs of care after birth. And this can really be done more effectively very often before birth, before a family has gone through a delivery, before they have a newborn in the NICU. When they're still in that prenatal space, they might have more bandwidth and time to be able to engage in some of these topics. Also, a goal is to provide couples with information to optimize their outcomes based on their personal values and preferences, and that's really important to us. But all these changes in medical management really require timely and certain genetic interpretations. And I think this group understands that very clearly. And some cases that we see when we do sequencing are very clear cut and very much very clear. So in this case, this is a fetus that has severe micromelia, a narrow chest cavity that could possibly be lethal, as well as an increased nuchal translucency. And what we found in this case was a de novo glycine change in the COL2A1 gene associated with achondrogenesis. So this was definitely related to the phenotype. Missense is a known mechanism for disease, especially glycine changes. This was classified as pathogenic. And this really allowed clear prognostic information and recurrence risk to be given to the family and impact clinical management. But many cases are not so clear. And as we know, genetics and genomics is very complex. We're able to generate a great deal of data, but we still have a lot of questions regarding how to interpret that data. And so through the years, since we've been sequencing since 2015, we've really appreciated that curating fetal genomic variants has some unique challenges. And of course, determining the pathogenicity of any given variant in the genome requires accurate phenotyping, as well as bioinformatic resources and expertise. So some of the unique challenges that we face in prenatal sequencing interpretation are listed on this slide. For instance, we have incomplete phenotypic fetal data due to the limitations of ultrasound and the temporal limits inherent to fetal development. We have a lack of data on prenatal phenotypes and genotypes in existing databases. And this may be because some of the most severe phenotypes are embryonic lethal or cause demise and are therefore never tested or documented. We also don't have any specific repositories for that type of information at this point in time. There also may be differences in the inheritance models between previously described childhood cases and fetal cases. I also want to point out that the data that we've been developing in this population from invasive procedures where we do testing on CVS, villi, and amniotic fluid is really informing the development of non-invasive screening techniques. So we will, I think, in the future eventually move away from having to do an invasive procedure, and we'll be able to provide all this genomic sort of information using non-invasive tools. So with all these challenges in mind, I want to pass it over to Dr. Ron Wapner, who's going to talk about some of the solutions that we've come up with to address some of these challenges.
Thank you very much, Jessica. I just want to continue by echoing what you say about the difficulty in phenotype, genotype correlations and a real understanding, particularly of many new phenotypes that we are seeing in utero that we've not seen previously in a pediatric cohort. Also, as you said, there's phenotype expression and variable expressivity, et cetera. And there's a giant need for us to have a resource in which we can gather enough information to make curating these situations easier. As with everything that we do, our willingness or our idea of starting a international repository for fetal genotypes and phenotypes started with a small group of us who, when about almost 3 or 4 years ago, we were really just beginning to use sequencing as a translational clinical tool. And we were very concerned that we needed to understand all the implications of this before it went into general clinical use. So we started having a conference, less a conference and more of a collaborative call between a few centers, and we all presented our cases so that we could learn a lot about what's necessary to translate sequencing to the in utero fetus. Those conferences, in my opinion, were incredibly valuable. We learned many, many lessons. I think one of the lessons we learned is that sequencing is not one person's game, that in order to really understand it, we needed teams. And so we then began to add laboratory people when necessary, content individuals. We already had MFM and genetic counselors, and the group expanded. That group then expanded further through absolutely word of mouth. Other people, as they began doing prenatal sequencing, either said, can we join and present our cases? Or we actually kind of followed the technology and where it got started. This relatively informal discussion expanded to become more formally the Fetal Sequencing Consortium. And we've listed some of the centers that are participating, but it now includes greater than 20 national and international groups. And again, it's a combination of disciplines, which is exactly what makes it valuable. And what I didn't say, we also have a number of pathologists who do autopsies on these cases. And equally, if not more important, we've now had a number of basic science geneticists, genomicists join us to help with the clinical laboratory correlation. So I would say right now we probably have more than 30 centers, and our biweekly calls have now had an audience of over 50 individuals. And as you can see, they've come from all over the world. We recently, in the interest of the world being so big, started a second fetal sequencing consortium for the other side of the world, predominantly based on the interest of the Australians. So once a month, we also have the Eastern Hemisphere with us. So it really has expanded dramatically, and the attendance has continued to grow. From this, we have obviously collected a large number of phenotypes and genotypes, have been able to do that on a case-by-case basis, and we've now gone through 3,000 anomalous cases. The collaboration and cooperation of this group has been phenomenal. We have published a number of cases on a number of different genotypes. We also have a group of us that have now recently been accepted as a center in ClinGen, and our goal in ClinGen is to evaluate or curate severe congenital anomalies and stillbirths, at least as a start. So we're pretty amazed and happy of how this has expanded. But we now are to the point where we have to make this usable data. It's been very interesting on a personal basis, and we learned what we needed to learn, but we now needed to make a resource that would be available to the community. So our next step, which we've started just recently, is to build a harmonized central repository that will be available for both clinicians and also for researchers. So the next slide illustrates the basic principles and I apologize for such a busy slide. I'll try to break it down, but each of these are important parts. And there were two basic premises that we wanted to use when we started this. Number one, there are lots of people in multiple other fields that have also undergone building repositories. So we didn't want to reinvent the wheel in any way. And we also didn't want to make the same mistakes that other people had made. So through the collaboration with newborn screening, through Amy, collaboration with the GA4GH, collaboration with some of the individuals that have made the tools that have fed these repositories, we think that we've gotten to the point where we can now test this. And how's this going to work? First of all, we picked 5 pilot test centers. The first 3 are part of our ongoing NIH-funded study to sequence and look at the clinical utility. But we also added another site that is not in any funded trial to see how this would work in routine practice. And we also thought that if this is going to be valuable, we need to see how industry will be able to utilize this tool too. So we actually chose an industry partner who has been doing prenatal sequencing for a long period of time. Our first step was to harmonize the data because everybody was using their own pipelines. Everybody was basically doing things independently. And for a repository, there needed to be some standardization. We have developed, and with the contribution from Illumina, a single system. So all the sequences that are coming from multiple sites are going to be run through a single system and harmonized. That's on the genotypes. The other thing, and I think this is really more important because in the world, so to speak, genotyping has run way ahead of phenotyping, and we've been led by Peter Robinson, who will talk more about how we're going to harmonize the phenotypes into a number of different ways. So that this will be, first of all the data will be harmonized. It then will be put into a common repository. As I said, that'll be harmonized genomic data, and we've already started doing that by collecting the FASTQ files and running them through a single system. That doesn't mean that system is any better. It doesn't mean that system is any worse. It's just we wanted to standardize things and hopefully also bring some information about, what leads to the best pipelines in fetal evaluation. And again, the harmonized data will come through phenopackets or phenotypes that David will talk about. Each of these processes have been using the de-identification and the data transfer, et cetera, from GA4GH, and they've been incredibly helpful in helping us transfer data into a single system, because our goal, and as many of you are aware, getting data from multiple institutions is not easy, and getting data from institutions outside the United States is difficult. We also made the decision that this should be a federated system because everybody is somewhat sensitive to transferring their data. So all the data will be entered locally, will be owned by the local provider, but will reside in the cloud also. But there will be a steering committee that will then decide how that data is used and which data can be utilized. So those were the basic principles. So we're working on this. We also have fortunately and thankfully been given existing tools that people have already used in the ongoing studies, such as All of Us and a number of others. Finally, we are using the FAIR principle that NIH has had multiple workshops on and have really suggested that anybody that is going to build a repository at this period of time should follow those. And that means data should be findable, it should be accessible, it should be interoperable, and it should be reusable. And much of what we're doing, and I think particularly when you hear Peter talk, the phenotypes, in order to be particularly reusable, and I think that's code for we can have a machine evaluate it or a computer, they all will be broken into HPO terms, etc. Bottom line, we still have a heck of a lot to learn, but we're moving forward with this. And only through wonderful partners that have been working with us have we been able to get this to a point where it's a reality. With that being said, I'd like to turn this over to Peter, who will talk a little bit about the phenotypic component of what we're doing. One last comment in case it doesn't come up in the questions. We are very interested and think, and that's why we love this meeting, that neonates to us are just slightly older fetuses, that some homogenation of the neonatal data and the prenatal data would actually be very important. Sorry, Peter, to steal 30 seconds of your time.
Thanks, Ron. Okay, well, thanks very much, Ron and Jess. I'm going to present very briefly the human phenotype ontology and discuss what can be done with such an ontology to support genomic diagnostics. I'm going to save information about our recent efforts to extend the human phenotype ontology for prenatal phenotypes to the question and answer period. Just suffice to say that together with Ron and Jess, we've assembled a working group of, I believe, about 70 people who are helping us to extend the HPO with new terms and new disease annotations. That effort is just getting started. So what is an ontology? Very briefly, an ontology has two use cases. One of them is pretty simple. It's just about exchanging data. And it's not a very difficult topic to understand if you're using two different words or two different codes to refer to the same thing in two different databases. Well, it's hard to exchange data. And an ontology can be used as a standard. And if everybody uses the same standard to refer to the same things, then it becomes easier to exchange data. The interesting part of ontologies is perhaps to act as a foundation for sophisticated bioinformatics algorithms. And ontologies are models of specific domains of knowledge. In this graphic on the right-hand side, I'm showing you an ontology-based model of Fanconi anemia. And so we see in the top left a node for Fanconi anemia that is connected by a has phenotype relation to 6 phenotype nodes. Those are human phenotype ontology or HPO terms. Obviously, there are many more for Fanconi anemia. Some of these phenotypes are shared by other diseases, and other ontology nodes reflect concepts from the environment, genetic subtypes, modes of inheritance, and genetic functions, and so on. And so together, many of us in the open source ontology scene are collaborating to make our ontologies work together to provide a harmonious model of medicine and many other domains. The human phenotype ontology, we developed this in my group in 2007. And when we published this in 2008, we were contacted by a number of other clinical groups who said, great, we've always wanted an ontology like this, but it's not good enough yet. Can we help you to improve it? And that started a tradition of workshops, and we've held, roughly speaking, 50 workshops over the last 12 years that have been between an afternoon and a week with various domains in medicine. And the workshops that we're having now are the first for prenatal medicine. And so the HPO has become quite a community project, and many of these papers have many co-authors from the community who have kindly given their time and knowledge to improve the HPO. And so the HPO has become unique in I guess 2 main ways. One is that it's by far the most comprehensive ontology of phenotypic features. There are currently actually over 16,000 terms. That's roughly speaking 4 times more than any of the sum of all of the other relevant ontologies in the United Medical Language System. The other thing is that the HPO is a sophisticated computational tool. And so what we see here in this hierarchy is that, the top node is just a very general node representing phenotypic abnormality, and the ontology branches out into different organ systems, and the organ systems branch out into the sub-organ systems, into morphology and physiology or function. And as we go down the ontology, we get to more and more specific terms. So for instance, deeply set eye is a specific kind of abnormal globe location, which is a specific kind of abnormal eye morphology, and so on. And the terms are themselves are defined computationally with reference to other ontologies for anatomy, function, histology, embryology, and a few others, such that immediately the HPO is linked to knowledge about these fields. So what do we mean when we say phenotype? Well, actually, we should have called the HPO the human phenotypic feature ontology. The word phenotype itself is used in a variety of contexts, but when we say phenotype, we mean a sign, a symptom, a lab finding, an imaging study, essentially the individual components of a disease. Two ways of making a diagnosis as a physician. One is a gestalt, so there are some diseases that we just recognize at first glance. Neurofibromatosis, Down syndrome, Marfan syndrome, sometimes can be recognized in this way. But given that there are over 10,000 named rare diseases and probably 20,000 altogether, it's impossible to gestalt all of these diseases. And so most people will write down the individual findings in this way. And this is something that we've called computational deep phenotyping. And this is the main thing that the HPO is designed to support. HPO has been used for many different things. You can use the terms of the HPO to describe a patient that you're seeing. You can enter these terms into software and get back a list of differential diagnoses. This has been particularly powerful in combination with exome or genome sequencing. And so we've developed a tool called Exomizer that's freely available. This has been used in many different contexts, including the 100,000 Genomes project in England, and also in the NHS there. The HPO has been used for translational bioinformatics research to essentially correlate genomic features such as tag boundaries here with phenotypes and diseases. And HPO is increasingly being used in the electronic healthcare record environment. So I'll stop there and be happy to take any questions about our plans for extending HPO to prenatal medicine during the Q&A session.
Thank you so much, Dr. Wachner and Dr. Robinson. With that, we're going to move to our final objective and really thinking about our opportunities for collaboration with the newborn screening networks and our colleagues. And so one thing I just want to say is that really the world has changed right in front of us. We're able to diagnose many more conditions in utero, primarily conditions that are not represented on newborn screening at this time. So I think this intersection between what we're doing in the prenatal space is really important to inform what's going to be happening in newborn screening moving forward. Clinical utility of genomics in prenatal care and management is really expanding, and our understanding of that is evolving, and there's a number of large studies that are looking at that. We can expect data on that soon. And additionally, our work in prenatal diagnosis across a variety of different technologies is informing further development of non-invasive techniques. So all of this testing that we're doing will be available, I think, in the future in some capacity using non-invasive tools, and then more readily available to more families. But overall, we do notice a lot of different challenges for further expansion of the use of genomics. And a big one that we've spoken about today is the lack of centralized prenatal data, both genomic and phenotypic. And what we've shared today are some ideas for how we think we can solve around that problem, and how solving that problem can really help the newborn screening space as well. We will continue to think about technical challenges, what conditions to be including in expanded screening, how to interpret variants, and what to report, and so on. So in terms of collaboration, sequencing in the prenatal period, I hope it is clear to you after this talk today, really can help better inform the postnatal management. And data from both prenatal and postnatal exome and genomes will allow for more robust, efficient, and accurate diagnoses in both time periods, really improving clinical care across the continuum. And so what we want to do together is leverage the NBS-TRN and team's experiences to develop a registry that incorporates the genotypes or genomic information and phenotypes for prenatal cases with congenital anomalies. And as Dr. Wapner pointed out, we want to make this available to both clinicians and researchers. And really working together, we want to develop a longitudinal follow-up to better define the genotype-phenotype correlations, and overall work together to support genomic sequencing information across the lifespan in collaboration with all of you. So with that, I want to thank you. Thank you for your time and attention, as well as the invitation to speak today. If you have questions or interested in joining our fetal consortium, please feel free to email me. My email is listed there. And now this is open for questions today. Looking forward to hearing from you. Thank you.